All right. Fantastic introduction, Michael. I feel like Michael just covered everything that I'd like to talk about, but in like a much richer detail. So this will be like the kind of kindergarten version of what Michael just talked about. So as Michael alluded to, we kind of have this unspoken problem as whole brain neuroimagers, which is that we want to make contact with the clicker that's not working. So I'll do this. So we've got these neurons, these synapses deep in the brain that are part of larger cells. I'm showing you here a PT-type pyramidal cell from the cortex. They themselves are a part of a complex cortical column with multiple different types of excitatory and inhibitory neurons, which themselves form these masses of columns that are interdigitated across the cerebral cortex. But don't forget, they've also got cortical inputs coming from all over the brain, thalamic cortical inputs coming from the thalamus, and then also neuromodulation coming from the brainstem and forebrain. These are then interconnected into this massive unit of sort of architecture that's spread across the brain, across multiple systems. And yet we're studying these things at this high level of the brain, and we'd like to be able to make contact between them. And I think, I don't know if you find Michael's argument as compelling as I do, but I think we really can do that if we start to think about the brain imaging experiments that we have, the results that we have, in light of these ideas of modulating neural gain and changing neural excitability with neuromodulators. So we're essentially left with this problem, right? We have to, how do we just analyze all this distributed activity that we get, these tiny little blips of activity that pop up across the brain? So rather fortunately, the brain is relatively low-dimensional. So what do I mean by that? I mean that of all of the potential degrees of freedom that the brain has, of all the billions of cells that could be connected by trillions of connections, the brain doesn't express all of them. A certain neuron activating is bringing other neurons along for the ride. So what we do is we have this sort of like degenerate code across our brain that we sort of utilize this sort of low dimensionality to embrace the world and all of its complexity and trying to learn how to adapt to it. By way of analogy, I think this is quite this is helpful for me. If you imagine taking a Rubik's Cube and trying to take a single square on the Rubik's Cube and turn it without affecting a single other square on the Rubik's Cube. And if you think about it for half a second, it's actually impossible, right? Anytime you flick the Rubik's Cube in any one direction, you're affecting both the target of your flick, but also all of the other stickers that are on that particular element of the Rubik's Cube. So in essence, this is the kind of crux of low dimensionality, that by doing some action on a system, you're bringing other elements of the system along for the ride. So what if we could measure this? That would be fantastic, right? So we can't measure it directly, but we can make estimates. So one way that I really like to use is called principal component analysis. It's not the only way to do this, and there's lots of other really cool ways being invented every day, it seems, if you follow arXiv. But the basic intuition here is that if we've got two regions, let's say we've got a region 1 and 2 plotted on the X and Y axis, and they've got some obvious correlation between them. What you can do with principal component analysis is try to fit a line of best fit. So this is you can either think of this as maximizing the variance across this population, or minimizing the residuals from that line of best fit. These are essentially equivalent. What you can then do is then after you've done that first line, you then look for another line which is orthogonal to that first line. It can be orthogonal in any of the remaining dimensions, and you again try to maximize the variance or minimize the residuals, and you do this so on and so forth until you think you've got an accurate representation of the data cloud that you have. Principal component analysis is a linear approach to matrix factorization, and there's lots of other really cool ways to do this. But what you then do is you throw out the original axes that you had, so we have to forget about region 1 and region 2, and then we reorient our data to this new axis. And so essentially what we've now got is a new X, Y, Z, all the way up to N coordinate system in which we can track the activity of our brain or our network of interest without focusing on one particular region of interest. So you may have heard this described as something like a low-dimensional manifold. This gets beat up on Twitter quite a bit because it sort of sounds like a bit of a silly word. But really what it's saying is that there are low-dimensional constraints on the activity of an otherwise high-dimensional system. So if people are interested, this is absolutely a beautiful paper by Manuel Zimmer's group from 2015. What they did was took a C. elegans worm, where we know an awful lot about its brain. We know what each neuron is, what we think it's doing. They took calcium transients from each of the neurons, and instead of looking as if from the top down on this city of fireworks popping off, they basically fit a PCA to that data. And what they found was that you could actually track what the worm was doing depending on where it lived on this low-dimensional manifold. So as you express these different degrees of freedom in this low-dimensional subspace of activity, you can actually tell something about the behavior of a worm. And so this was super cool, really beautiful paper. And we started talking about this. I was having a chat with Olaf Spons, who was one of the collaborators on this project, about whether we could look to find similar kinds of things in the human being. Now, one of the challenges is that the Zimmer group were able to actually capture the C. elegans doing pretty much what it likes to do, which is to wiggle forward and backwards and then turn around and turn right and then stop. And so we wanted to try to capture something like the breadth of human cognition. We did the next best thing, which was to use the Human Connectome project. Here we've got 7 different tasks that are purported to try to capture all of the different sort of functional capacities, or sort of at least a large spread of these. So for those that are unaware, there's these 7 different tasks that range from emotion recognition, a gambling task, there's a math and a language task, a motor task, an NBAC cognitive load task, a relational matching task, and a social task. And so what we did was we sort of put all of these together, we appended them over time and across subjects, and what we wanted to do was to fit a principal component to this analysis, to this data, to see if we could find a low-dimensional architecture within the human brain. And so lo and behold, it's precisely what we found. So with even 5 principal components, we could explain 70% of the variance in the data. What that means is that with just 5 different dimensions, instead of the 400 that we started with, we can collapse it down. So we go from voxels to regions of interest, and then down to these components. We can explain a lot of the variability in the brain over time as a function of these different tasks. One of the things you get out of a principal component analysis is a set of spatial maps. So what you can essentially do is take the weighting of each of the principal components across all the regions of interest and plot them onto the cortex, subcortex, and cerebellum. So for example, the first principal component, which explained about 40% of the variance in our study, was loaded onto frontoparietal cortices as well as visual cortex, striatum thalamus, and superior cerebellum. And then you saw a negative loading onto areas like the default mode, the nucleus accumbens, hippocampus, amygdala. And you can see there's a sort of different pattern that's quite heterogeneous across these different PCs. I remember when I was first talking to Michael about this, when we got some of the initial results, I was quite excited, like, oh, look, you can see the default mode in there. And Michael asked me, like, you're not thinking about this like intrinsic connectivity networks, are you? And I remember like, no, of course not. Like, why would I think about it like that? And the reason he said that is that PC dimensions are not like the traditional network where you grab seed to voxel maps and then you track them over time, their engagement. Rather, they're kind of like ideal situations that you could get to. They're like directions on a landscape through which the brain can evolve. So I think there's a really nice framework for thinking about this, which is the idea of an attractor landscape. So this is quite common in physics, particularly mean field reductions. The idea is that you imagine that the state of interest, in this case the brain, is an element that's rolling around a landscape, and it follows essentially gravity. So in this case, this brain state will fall down into an attractor well, and you'll be in that next state. One really fascinating thing is that neuromodulators like noradrenaline and serotonin flatten the attractor landscape, so they essentially make it much easier for you to get into otherwise distant attractor wells, and then you can form a new attractor and head into that new brain state. So this is a sort of a framework for thinking about this low-dimensional system and its evolution. So with that in mind, I'll show you here a video of the first principal component and the second principal component. Every dot will be a TR, and they're all linked together in time. They'll have a little line between them, and they'll be colored according to which task they were in. So what you can see as this plays out over time is that you can imagine that the brain state here is rolling around a landscape where an attractor will form on the right side of the screen, and then it'll dissolve and bring you back into an attractor on the left side of the screen. So just in that there really is this low-dimensional sort of structure to the flow of the brain over time. And a couple things to notice is that you do tend to go to very similar places as a function of task, but not exactly the same places. And the second thing is that there are large portions of this state space where the brain just doesn't seem to go, at least in this context. So there are, if you're trying to load down here on low on TPC1 and TPC2, you simply won't do that in this context. So this is really suggestive of what we would call a sort of low-dimensional manifold. So another thing that we noticed quite early on, so this is just a cartoon of how you would create these time series. You take the time series of interest, which have a particular loading for the different PCs. You then create a weighted sum, and you plot that weighted sum over time. So this here is the eigenvalue for the eigenvector of the first principal component. And if you relate that to the task block structure, there's a really remarkable overlap. We ran a number of different permutation tests to convince ourselves that this was in fact a pattern in the data, and it looks like it's a really robust effect where being in a task block pulls the state into one configuration, and then being in a rest block pulls you out of that configuration. So what you can then do is you can try to estimate from that this sort of low-dimensional manifold, the sort of patterns of activity that are allowable in the system. What we could do to convince ourselves that this was actually useful is compare it to things like the Neurosynth database. So it turns out that being on the right side of this manifold is associated with motor and cognitive tasks from the Neurosynth topic analysis, and the left side of the manifold is associated with memory and language functions. And you can do a similar thing with brain network architecture. So we found that the brain was more integrated on the right side of this manifold and more segregated on the other side of the manifold. So just think that there really is this sort of low-dimensional axis that can pull you into and out of different cognitive states that have a different distinct topology. Again, you'll notice a theme. I quite like analogies. This is the ball rolling around the landscape, pulling you into, say, a memory state before you do some cognitive work on a task, and then finally make the decision to push a button. So I really like this analogy for thinking about it. But it still leaves us with this problem, right? Like, what is causing these fluctuations? Is it just something like the task structure, or is there something else about the sort of architecture of the brain, the biological architecture, that's driving these fluctuations over time? So if anyone saw Katie's awesome keynote last night, you'll know that there's this sort of emerging idea that the ascending arousal system, the basal forebrain, that she showed some beautiful work from her neuron paper, but also things like the noradrenergic locus coeruleus, the dorsal raphe projecting serotonin, and many, many more, they have this sort of low-dimensional control or shaping function over whole brain dynamics. And so we wanted to test this idea. To do so, we leveraged the Allen Brain Atlas, which is an open resource where they've taken 6 postmortem brains and they've performed gene analysis inside in essentially like a searchlight fashion across the brain to try and quantify the amount of each of the different genes in the human genome in each of the different portions of the human brain. Instead of looking at every single gene, we decided to collapse this down to a set of genes that related to neurotransmitter receptor profiles. So the idea here is to identify noradrenergic, dopaminergic, cholinergic, and serotonergic receptors and to work out how they relate to the manifold. We focused on metabotropic receptors because ionotropic receptors bring a whole bunch of other questions that come with them, but there's also fascinating things I'd love to talk to people about later. But so the idea was to try and take these different receptors and to put them into a couple different groups. Based on the literature, we identified that alpha-2A receptors, things like guanfacine will activate that, promote things like working memory, dopamine 1 receptors, M1 receptors, and 5-HT2A receptors. You'll hear a bit more about what happens if you over-augment the 2A receptors a bit later from Joanna. And then neurotransmitter receptors that can diminish cognition, so things like the alpha-1A receptor, dopamine 2, and 5-HT1A. The literature's a little bit murkier for the muscarinic receptors, so we didn't include one in the cholinergic group. And so here what I'm showing you now is the TPC1 and TPC2 axis, so our original embedding space. Every grey dot is essentially the results of a null model when we compared the receptor maps to the original manifold to see whereabouts the receptor maps would pull you into this embedding space. And essentially what we found was that the neurotransmitter receptor profiles that augment cognition were associated with the right-hand side of this manifold. The side I told you was associated with cognition, motor performance, and an integrated brain. And on the other hand, you have the other receptors like D2, alpha-1A, and 5-HT1A that are associated with pulling you out of a cognitive state into a more memory or language-based state and to a more segregated architecture. So in the last couple of minutes, I want to sort of do a little bit more speculating. This is absolutely fascinating to me when you think about some of the projections that come from the lateral prefrontal cortex and the orbitofrontal cortex. So if you read the literature, it turns out that there are unique areas within the lateral prefrontal cortex and the orbitofrontal cortex, as well as the anterior cingulate, that project down to these individual brainstem and forebrain regions that in turn augment neurotransmission back in the cortex, thalamus, and cerebellum. So here I'm showing you the basal nucleus of Maynard, which is cholinergic, the ventral tegmental area, dopaminergic, and then the raphe in purple, which is serotonergic, and the locus coeruleus in red, which is noradrenergic. And so the idea here is that you've got these projections that come down to these different brainstem and forebrain regions, which can then in turn, based on context, decide how much of each of those different neurotransmitters you'll pump back up into the brain, which will then lead to changes in the gain modulation in specific patterns of brain network architecture. So I think this is actually really cool, and you can think of like an analogy here, right? Like there's essentially like a set of controls in the frontal cortex now. You can pull the volume up on this bit, you can tweak the knob on that one, and you can pull yourself into ever more subtle parts of state space by controlling the gain of a different set of systems using this balance of neurotransmitters. I think this is particularly fascinating when you think about the fact that the lateral cerebral cortex, along with the lateral cerebellum, has expanded over evolutionary time. And so if we can take that pilot analogy one step further, it's almost like as evolution has progressed, what we have is we're going from a set of relatively simple controls where you can be particularly motivated or excited about something, to being motivated but not particularly excited, or excited but really kind of distractible. And so I think there's like this idea of a much more subtle kind of architecture of control as we go through evolutionary time. And with that, I'll say thank you very much.